Hello everyone, welcome back to my channel. I am Tepin and today we'll be learning about how to process newly generated DNA sequences using the given software and also generate consensus sequence that can be used for further phylogenetic analysis. So these are some sequences that I have. This is ITS5 and ITS4. So before starting, I would like to mention specifically this one software called Bio Edit. Insert Bio-Edit software, then download it from here. You can download it. Download now. You can download like this and then open it, extract it, and then install the software. Search Bio-Edit. You will find something like this new shortcut or something. Or you can also check the software here. You can see here all. You can go through here. Here you can see it is new shortcut. Or in some system you will find it like Bio-Edit and like that. So after downloading and installing the software, you have to do one thing. For example, in some cases your file will not look like this. As you can see here, it is green in color. So you have to go check this AB1 file here and then change the properties and change the open width. You can see open width, change it to a Bio-Edit sequence alignment editor. If you cannot find it here, you can browse the app. So you can choose an app on your PC and then here we can see not in program file, you will find it here. Bio-Edit. You can choose this Bio-Edit and then set default, apply and close. Okay, and then your all the AB1 file you will find it like this one. It will turn into green, this Bio-Edit sign. Then you just have to double click this one and you will see something like this. This is a good sequence that I've already made a video on, which are good and which are bad sequences. This is a very good sequence with very good peak, clear peak, no noise. You can keep it like this. And then press Ctrl O and choose your other sequence. For example, initially I opened this ITS5 forward and this ITS4 in the reverse. You have to open this one also, and you can see this one is here. This is also a very good sequence. And you can close this one, everyone. Okay, close this one, no, and then click here. This is your ITS5 forward. Click here and then press Ctrl plus I. Okay, it's not working, so you can just go to file. Don't forget to click here. Okay, click here first, this ITS5 file, and then import sequence file alignment, and this is the file. You have to choose ITS4. So you can see this is your ITS4, this is your ITS4, and this is your ITS5. I have imported this here. Now since forward and reverse are reverse complementary, so you have to make sure your reverse sequence is complementary to forward sequence, not reverse complement. So in order to change it from reverse complement to complement, you have to press Ctrl Shift R. So this is now complement with your ITS4. When you will align this one, just click here don't choose anything. If you choose like this, it is not going to work. See, okay. So you have to either choose all or just not choose anything. And then go to accessory application, cluster W multiple alignment, run, okay, and wait for a few seconds. So this is the aligned file. You see, this is completely aligned. If your file is like this, for example, it is aligned very properly. You can see, you can click this one to see your sequence. You can click here to see if there is any changes between the two sequences. So you can see it is aligning from here very, I mean, this is aligning very good from here. You can just delete this one, then go from here and check if there is anything, any differences between the two sequences. You see, there's no differences. There's one gap here, but other than that, it's all good. You can remove from here also if you don't want. If you're not very sure about anything, you can just delete this one and then go to alignment and then here create consensus sequence. So it is going to create a consensus sequence. All that. You just copy from here. Uh, okay, sorry, I forgot to mention few things. How am I selecting this? First click here, then select and then press shift and then click here. So it will select like this. Click here and then click here, pressing the shift. Click here, press shift and then click here to select it and then press delete to delete. If your delete is not working, it must be probably because the mode is not in edit mode. It might be in select mode. So you have to change it to edit mode if in case if you are not able to delete the sequence like me. And then go to the end, click here, press shift and click here and then press delete. And then go to accessory application, sorry, alignment and then create consensus sequence. Okay, so it is not working because we have to align it again. If consensus sequence is not generated, you can align again. And the thing is that you can see consensus sequence is 100% similar to the forward sequence. So in case if your sequence is totally 100% similar, the forward and reverse sequence is 100% similar, you can just copy the forward sequence because forward sequence will be the consensus sequence. You see, they're all similar. There's no difference. You can just either create consensus or you don't have to do that. Just select the forward sequence, shift and then click and then press Ctrl C, copy and then make a new file, ITS. Since you have merged both ITS 5 and ITS 4, so this is your complete ITS region. Not complete ITS region, I mean uh, a consensus sequence generated using both forward and reverse sequence. So you can just save it like, for example, this is Hymenopete and this is ITS region. And for example, your SSN number, you can put ARF 200 just for example, and then you can say ITS and Control S save. So this is the ITS region. You can go to file and then save as, and then click here and then dot FAAS, all types, then save. This is your FASTA file. See, I'm looking at ATS. This sequence you can use in your phylogenetic analysis or any kind of analysis. You can also submit this one because this is completely reliable uh, sequence which you can submit. So this is it. Thanks for watching, my friends, and don't forget to hit like, share, and subscribe. And I'll see you in the next video.